Well documented as a botanical medicine for over 5,000 years, the discovery of endogenous cannabinoid receptors and evidence-based reports indicating the efficacy of medical cannabis for treating pain have renewed consideration of its therapeutic use. The past quarter-century's advances in healthcare science has included the discovery of a previously unknown human endocannabinoid system dedicated to maintaining critical balances between our mind and body. The following recording derives from a 6-part series of sequential lectures concerning endocannabinoid receptors and medical cannabis, as narrated by physician expert and author Dr. Jeffrey Block. This publication emphasizes how an appreciation of the biologic context to show how and why endogenous cannabinoid receptors function allows healthcare providers to better interpret the limited evidence-based data. In order to properly assess the abundance of anecdotal reports concerning cannabis, the safe and effective treatment of pain will require careful clinical monitoring of patient outcomes to establish and validate the evolving evidence-based data. Dr. Block is an American board-certified anesthesiologist and respected thought leader with a clinical scientist's understanding of the healing potential found with certain botanical medicines. Fellowship trained in both pain management and addiction medicine, his pragmatic rationale considers a citizen patient's compassionate access to the cannabis plant's bioactive chemicals when used for the treatment of pain, as well as acknowledging cannabis's obvious public health concerns ranging from contaminants to dependence. The author's insight shares contemporary perspectives by first acknowledging the bioethical obligation healers have to not simply feel compassion for their patients, but to strive to effectively treat pain. This is emphasized while at the same time acknowledging those concerns to public health surrounding addiction and accidental overdose deaths. Dr. Block's writing reveals how and why a human being's natural instinct nurtures their nature through their body's own endocannabinoids, and why a contemporary physician healer's understanding of the traditional ethnobotanical role certain plant-based medicines have played in treating the intolerable symptoms of disease appears to have also tangible value in modern healthcare. As read from Endocannabinoid Receptors and Medical Cannabis, here is the author's narration of Section 5, Clinical Considerations of Cannabinoid Medicines.
Dosing Guidance. Behavioral research has established that most patients have already experimented with using cannabis therapeutically before asking their physicians about it, if they ask at all. The concern patients have for how their doctor perceives them, coupled with the fear of being stigmatized as a marijuana user, indicates that patients using cannabis medicinally often fail to disclose that use to their doctors. The robust market in cannabis makes independent experimental use possible in ways that are not seen using conventional medications. As a result, it behooves clinicians to not just be prepared for such questions, but to be assertive about broaching it with patients for whom cannabis may be a legitimate treatment option. While some individuals may hide their therapeutic use of a stigmatized drug, others may ask physicians for medications outside the bounds of accepted treatment. Research indicates that 20% of individuals who present themselves in emergency room visits as having acute pain are seeking opioid medication for questionable or non-therapeutic reasons. Clinical guidelines for managing addiction liability include continuity of care and use of signed treatment agreements to describe and minimize risks, an approach that has been recommended for medical cannabis. Clinicians are rightfully concerned with providing accurate medication dosing directions for their patients. Though plants have been the source of treatments throughout most of human history, botanical medicines pose a challenge for accurate dosing because of the difficulty in obtaining reliably standardized products. The complexity and variability of botanical medicines contributed to the 20th century's turn away from plant remedies to pharmaceuticals based on isolated active ingredients that can be synthetically manufactured with consistency. This complexity and variability are particularly true of medicinal cannabis products and of the plant itself, all of which are highly variable in their chemical components. Even carefully cultivated cannabis plants of the same varietals, including clones from the same parent plant, may result in end product variability with differing cannabinoid and terpene profiles. This is multifactorial and can depend on a given plant flower's chemical variability, how and where it was grown, when it was harvested, and how it was dried and cured. Additionally, non-standardized chemical testing reports can vary significantly in assessing products, further complicating the matter. As with most medications, start low and go slow is a sound strategy for cannabinoid medicine therapies in most conditions. The variability of commonly available cannabis products is compounded and confounded by considerable variance in patient experiences based on inconsistent product types and routes of administration, use timing and routines, and physiologic set points. What counts as a suboptimal dose in one individual may be a robust effective treatment in another, and this often changes with individuals over time. Some of those differences may be associated with developed tolerance via receptor downregulation, but changes in sensitivity can affect some long-time users such that they report decreasing their dosing over time with comparable results. What serves as a low or high dose will vary between individuals based on that person's experience with cannabis use, their physiology, and the route of administration. Many state regulatory agencies have set dosing standards or serving sizes at 10 milligrams of THC, but doses as low as the clinical threshold of 2.5 milligrams of THC or 2.5 milligrams of balanced ratio THC to CBD products are commonly manufactured and used effectively by cannabis-naive patients. CBD dosing should be carefully integrated as part of a patient's overall pharmaceutical management because it can induce some cytochrome P450 enzymes, suggesting that it has clinical relevance concerning the breakdown of other medications, including blood thinners and other anxiolytics. Paradoxical dose-dependent effects on pain. For pain patients, care with dosing is especially important, as THC and whole plant preparations have been shown to exhibit dose-dependent effects that are paradoxically biphasic. Relatively low doses typically produce effective mild to moderate analgesia, but high doses can actually intensify pain sensations. This dose-dependent characteristic features a narrow window of effective dosing that may explain early clinical studies of cannabinoid analgesia that produced inconclusive results. The doses of THC vary between clinical trials, and the studies included in the National Academy's report produce some indication of dose-dependent effects. The importance of dose was also seen in studies of vaporized cannabis involving acute pain, but a similar study found analgesic effects not to be dose-dependent. Anecdotal reports of pain intensification typically entailed relatively large doses. Escalation in dosing can occur when a patient believes the amount taken was insufficient and subsequently takes more. That can happen when the time to onset of effect is underestimated, as is more common with edible cannabis products' slower GI absorption. Failure to achieve the desired level of analgesia can also contribute to a cannabis user surpassing the effective dose range as more and more is taken in hopes of achieving more relief. Increasing cannabinoid dosing may cause increased side effects, hence diminishing its overall therapeutic value. This is most profound with synthetic cannabinoids. When comparing synthetic cannabinoid medications such as dronabinol and nabilone, Many patients prefer the whole plant's balanced chemicals, critically describing the THC isolates as having all of the side effects without all of the benefits. At high doses, the effects of THC alone may not just be hyperalgesia, but acute anxiety and dysphoria as well. As mentioned earlier, CBD modulates these concerning effects of THC through direct and indirect mechanisms. Routes of administration. In addition to varietal differences, the effects of cannabinoid medicines can be dramatically altered by its route of administration, including inhalation, mucosal absorption, oral ingestion, and topical application. Smoking is the method most commonly employed by cannabis users, not only because of tradition and ease of use, but also because inhalation's speed of onset allows for quick symptom relief and effective titration management. It only takes a few seconds before the cannabis user perceives an initial effect and up to a few minutes until its peak effects are realized. The byproducts of smoke inhalation nonetheless make it an undesirable method. However, by heating plant material to a temperature below combustion but sufficient to aerosolize the volatile cannabinoid and terpene oils, vaporizing provides the benefits of inhalation's speed of onset without smoking's byproducts. Lessons learned from tobacco smoking's combustion release of cancer-causing benzene and polycyclic aromatic hydrocarbons remain a concern, although epidemiological studies have failed to find an association between cannabis smoking and the health risks associated with tobacco smoking, such as lung cancer, emphysema, and chronic obstructive pulmonary disease. Inhalation is well known to accelerate nicotine's central and systemic blood levels, but deference to the lungs' critical function of gas exchange has largely limited their use to deliver local therapeutic bronchodilators and anti-inflammatory medications. Nevertheless, when compared to combustion, vaporization's lower temperatures minimally adulterate the cannabis plant's bioactive component chemicals. While many vaporizers of cannabis oils and flowers are commercially available, devices that allow precise, consistent temperature control are ideal, as different temperatures will release varying combinations of cannabinoids and terpenes. Some experienced cannabis users may report reduced effectiveness because vaporizing lacks cues associated with smoking that may produce an independent placebo effect, but vaporizing appears to deliver cannabinoids at least as well as smoking and better preserves a given varietal's terpene profile. Multiple clinical trials have shown vaporization to be an effective delivery method for pain management, and a crossover trial of vaporization and smoking found vaporized delivery produced higher blood THC levels and more acute effects at both low and moderate doses. Oral mucosal delivery methods are utilized for Sativex and Epidiolex, and other sprays and hard candies provide absorption through the mucous membranes of the mouth, with some content known to advance to absorption from digestion. Mucosal absorption offers superior speed of onset when compared with edible products, as well as a more systemic bodily effect than inhalation that delivers cannabinoids far more rapidly to the brain. Cannabis suppositories are also available and may be preferred by patients with persistent nausea, as well as those using higher doses that may not be well tolerated through edibles. Edible cannabis products come in many varieties, from infused chocolate and candies to cookies and sodas. Cannabis tinctures suspended in alcohol or glycerin, similar to those used in the last century, are widely available and can be added to food products or taken directly. Edible ingestion results not only in the longest time to onset, but also the longest duration of effect. The prolonged time to onset means that naive users may mistakenly believe that they have already experienced a full effect long before they actually do, and so they may then ingest even more cannabis. This is the most common way to an acutely uncomfortable experience that may induce intense paranoia and not infrequently brings an intoxicated, dysphoric patient to the emergency room. The consequences are compounded by the psychoactivity of 11-hydroxy-THC, the delta-9 THC metabolite produced by liver metabolism during digestion. Duration of effect from edibles lends to their use for overnight treatments when extended relief and sleep assistance are primary goals. Many pain patients who are cannabis savvy also report using low-dose edibles to maintain a baseline of analgesia with inhalation for breakthrough pain. Topical cannabis products are often combined with other therapeutic or aromatic creams and oils for treating arthritis or muscle pain. The evidence in support of topical application is limited to anecdotal reports, although some cannabis product manufacturers have been collecting data through individual and crowdsourcing in a more systemic fashion. As with all subjective reporting in the absence of objective clinical measures, caution is required in assessing the validity and reliability of this data. Of extra concern in the case of cannabinoid medicines are the potentially confounding effects of euphoria and memory impairment, which may individually or in tandem influence both the immediate experience and its recall. Risks and contraindications. The important role of clinicians in educating their adult patients about the potential risks involved in cannabis use needs to be emphasized when making it available for therapeutic purposes. Cannabis dependence and impairment are significant public health issues, but complications from cannabis use are poorly documented. Multiple animal studies of extreme cannabinoid exposure indicate at worst mild risks from its use. In humans, 3 primary relative contraindications for cannabis use are pre-existing or genetic predispositions to mental health disorders, heart problems, and pregnancy. Individuals with a variety of mental health disorders appear to gravitate to cannabis use because of a subjective perception of improved symptoms. Particularly in the naive user, higher doses, for example greater than 15 to 20 milligrams of THC, can induce feelings of panic and anxiety that can be problematic. Side effects of chronic cannabis intoxication, including performance declines, are more often seen in recreational consumers. Limited clinical research suggests cannabinoids may be effective in treating schizophrenia. However, the National Academy's report notes substantial evidence of a statistical association between cannabis use and the development of schizophrenia and other psychoses, and an increased rate of cannabis use is associated with a genetic predisposition to schizophrenia. Cannabis use consistently produces tachycardia and vascular dilation, which rarely may elevate the risk of a myocardial infarction in individuals with pre-existing heart problems. Those individuals with heart disease are more likely to develop chest pain after acutely consuming cannabis. While cannabis use can increase heart rate and systolic blood pressure, a decrease in peripheral vascular resistance can also result in orthostatic hypotension. Pregnancy contraindicates many medications, though, as with all individuals, physicians and patients should weigh the risks and benefits to arrive at clinical decisions. Lower birth weight is the most serious reported association with maternal cannabis use, but this may be explained by the prevalence of smoking as a delivery method and the concomitant carbon monoxide and carbon dioxide exposure that can create hypoxia in the fetus and limit growth. A study of neonates and infants in a population of heavy cannabis users found no developmental or other problems. Ethnobotanical use of cannabis by some Caribbean cultures has traditionally treated first trimester hyperemesis by drinking dilute cannabis teas to remedy the nutritional risks and restore the systemic fluid needs associated with this malady. Due to the ethical challenges associated with studying women during pregnancy, any potential consequences from cannabis use on fetal stem cells causing long-term effects are poorly understood. The antiemetic and appetite-stimulating properties of cannabis has been reported to benefit women afflicted with severe morning sickness with fewer side effects than other medications. But should women suffering from morning sickness during pregnancy use marijuana to control their nausea? In states with medical cannabis programs, health experts, regulatory officials, and most patient advocates have consistently answered no in the form of public service announcements and warning labels on cannabis packaging. Nevertheless, a statewide cross-sectional study in Colorado found that nearly 70% of dispensary workers endorsed using cannabis for first trimester morning sickness, and fewer than a third spontaneously recommended consulting a healthcare professional about it. In contrast to morning sickness in gravid women, cannabis hyperemesis syndrome is a rare but serious adverse event associated with chronic cannabis use. This is a paradoxical condition in which an individual with consistent long-term exposure to cannabis may suddenly develop uncontrollable vomiting following cannabis use. This effect is striking given the well-documented and far more common antiemetic effects of cannabis use. Symptoms are reportedly often relieved by a hot shower or bath. While the etiology of the cannabis hyperemesis syndrome remains unclear, cannabinoids are known to inhibit calcium channels, and calcium channel blocking is associated with nausea. Furthermore, calcium channels are temperature sensitive, and heat increases channel firing that mitigates nausea, which may explain why afflicted patients often report that a hot shower or bath relieves their hyperemesis. The paradoxical nature of the syndrome and its rarity even among populations of heavy chronic users suggests factors other than plant constituents, including an autoimmune phenomenon, may be responsible. Another explanation posited by some knowledgeable with cannabis cultivation practices is chronic exposure to azadirachtin, the active chemical compound in the biopesticide neem oil, which is commonly used in organic farming. Contaminants and Testing. Pesticides of all types are a concern with cannabis cultivation, and inadequate regulation and testing in some U.S. state markets has resulted in cultivators using chemical insecticides and fungicides on their cannabis crops. A random testing of cannabis commercially available in California and Washington legal markets detected residual pesticide contaminants with known potential for serious health effects in a majority of analyzed samples. Health Canada requires testing for pesticides in the medical cannabis distributed through their program and conducts its own random testing of products. In 2018, 2 Canadian suppliers were required to recall all of their pesticide-contaminated products. In recent years, many U.S. states have adopted regulations for what can be used safely in cannabis products intended for human consumption and require testing of products. Due to federal prohibition, universal guidelines have not been developed, and organic certification standards are unavailable for cannabis cultivation. The absence of a United States Pharmacopoeia disposition on cannabis standards led the American Herbal Pharmacopoeia to issue a monograph on cannabis purity and identity standards in 2014. The American Herbal Products Association has similarly produced guidelines for regulators on cannabis products, including cultivation, storage, manufacturing, and dispensing. Both groups acted in collaboration with subject matter experts and patient advocates, and their standards have subsequently been widely adopted by certifying standards organizations and state agencies in the United States. Global standards organizations such as ASTM International, formerly known as the American Society for Testing and Materials, are also working to establish uniform protocols for analytic testing and other aspects of cannabis production. Life-threatening pulmonary complications subsequent to the increased use of vaporizing devices have recently been reported. While infectious diseases do not appear to be responsible, Other contaminants, including tocopherol, also known as vitamin E acetate, remain suspect and have been investigated as a source of chemical burns progressing to reactive airway pneumonitis. Additionally, diacetyl, a flavoring compound previously known to cause bronchiolitis obliterans in coffee and popcorn industry workers, is also suspect in a cannabis user's lungs subsequent to inhalation of this vaporized contaminant. Consistent state guidelines directing the enforcement of production standards have not been established while cannabis remains under federal prohibition. The Centers for Disease Control urge doctors to report suspected cases to their state health agencies, and the Food and Drug Administration is collecting information about illnesses related to e-cigarettes and vaping. The broad range of endocannabinoid system effects requires caution in pharmacological applications. The effects of long-term chronic use of exogenous cannabinoids on the system's function is unknown. However, limited studies of medical cannabis users enrolled for more than 30 years in the federal government's investigational new drug program have found no adverse health consequences. Possible downregulation of endogenous cannabinoid receptors, such as is seen in other receptor systems, requires longitudinal studies to determine significance, but this may be a neuroadaptive factor of cannabis dependence. A 2012 investigation by the National Institute of Drug Abuse addressed the endocannabinoid system receptor's plasticity as measured by its ability to regenerate after chronic heavy exposure. Cannabis users sequestered without access to the drug had a near total recovery of their brain's full complement of CB1 receptors after only 4 weeks of monitored abstinence. Where significant patient outcome problems have been seen is with drugs that block cannabinoid receptor activity. The role of endocannabinoids in metabolism and appetite has made the endocannabinoid system a target for the development of drugs to treat metabolic syndrome. Rimonabant is one such drug that works as an inverse agonist at CB1 receptors to reduce hunger and weight gain, but at considerable cost to overall endocannabinoid tone. Although weight loss was documented with Rimonabant, serious adverse side effects included nausea, seizures, depression, and most importantly, suicidality, leading to withdrawal of the drug after 2 years on the foreign market. In this case, drug development failed to appreciate the full extent of the CB1 endocannabinoid receptor's broad range of nonspecific central effects.
This recording has been a healthcare educational initiative read by physician-author Dr. Jeffrey Block. Dr. Block is the founder of Nurturing Nature Group Consultants, an independent and scrupulously neutral interdisciplinary consultancy group advancing endocannabinoid science through translational research within the practice of integrative medicine. To learn more, please visit nurturingnature.com.